Okay, good day, everyone. Welcome to Science, Technology, and Society. Hello and welcome. I'm Sir Kenneth Boreal. Welcome to our asynchronous session for Science, Technology, and Society. Okay, so it's almost a few weeks to go before we'll end our course for Science, Technology, and Society, or STS. So this week we'll be discussing on the tenets of biotechnology, or specifically uh, genetic engineering or genetic modified organisms, or GMOs. So welcome for our students. Welcome to our students from the Georgie Ilagan College of Business and Tourism at the International Management School. So thank you so much for joining us on this session. So if you're just into uh, our video, our YouTube video, don't forget to hit the comments there on the chat box. Please place your name on the chat box so we can recognize you. If you're watching us on the web, it's over here. Or if you're using your phone, look for the live chat button under your video, under the video, and click there and type in your name in the chat box. Okay, so thank you so much for joining us. Today, we are uh, discussing uh, another specialized topic for SCS, GMOs, or genetically modified organisms, part of our module, GMOs and gene therapy. Okay, so at the end of this lesson, we'll share issues with GMOs, or genetically modified organisms, discuss different implications and the impacts of GMOs, health, and politics, and create a research paper on the impacts of GMOs in the Philippine context that will be discussed next meeting. Okay, some reminders before everything. Uh, Again, our reminders for the final requirement. So I hope everyone has grouped themselves, and and will be uh, creating their final requirement, which is a video, a mini documentary on science and technology issues. I hope everyone has already formulated their discussion on how they will present these different topics on the science and technology issues relating to tourism and hospitality. Okay, so hope you're still there uh, watching us on ano, deadlines will be posted on your Microsoft Teams account. I'll be posting the reminders for the final project within this week. Excuse me. Okay, let us discuss uh, a fair amount of slides. This is a fair amount of slides lang, just to introduce you to GMOs, genetically engineered organisms, and what are the implications on why people are still ecstatic on um, why is there such a thing as genetically modified organisms. So let us create a discussion on how to differentiate between what they call GMOs and genetically engineered organisms. What are they different, or are they the same? No? Let's start with our discussion of a GMO. No? For millennia, for thousands of years, um, we have already manipulate the DNA of plants and animals. Have you ever thought of that? No. So let's start first with one of the most important crops in the world today, corn or maize. corn was discovered, or maize was discovered in Mexico, and it was called teosinte, no? And as you can see, the previous look of this particular species looks like this. It's very quite small, no? Around the size of a dime, no? But with the development of modern agriculture, you can see now that through many years of developing of the species and selecting the best crops that this plant can do, now corn, modern corn or maize, has looked like this, like an ear of corn with all of these, one cob of corn with all of these ears. No? Amazing, pretty amazing for a crop like this, for a species like this. Let's look at this other illustration right here. This is the old version of a carrot. You know carrots? That one, this orange here. These were carrots in the ancient times. They were inedible, so to speak, but through the development of modern or through the development of ancient agriculture, carrots look like these orange things now. No? So can you ever imagine carrots look like this? Will you ever eat that? And come to look at here. This is a wild species of a tomato. Okay? This is the size. It looks like a little cherry or something like that. But look at the tomatoes today. They are huge in size. Tomatoes are grown to look like this now. No? Have you ever thought that we could have more of these tomatoes if we don't have uh, genetically modified all of these? Would you ever thought of that? Okay. Looks intriguing, right? Let me show you another. Oh no, let me show you another illustration. Okay, I know if you are a culinary or hospitality student, you know all of these. If you are, or if you are into food, if you're not a tourism or if you're not a culinary or hospitality student, you, you might not be familiar with these. But have you ever thought of these vegetables like cabbage, kale, broccoli, cauliflowers, and all of these arugula and uh, ano, all of these things here, artichokes, no, come from the same plant. It's just that they are the same species. Would you ever thought of that? Let us see. Actually, the plant itself, Brassica Alliance, the species, can be modified, can be selected for their different traits. For example, if you select Brassica Alliance for the terminal buds, it becomes a cabbage. If you select for lateral buds, you produce Brussels sprouts. If you use for the stem, you have the kohlrabi. If you have for the leaves, you have kale. If you have the stems and flowers, you have the cauliflower, or the broccoli rather. Or for the flower clusters, the cauliflower. It's the same species, but the plant itself has been modified to become these particular variants of the species. Okay, so they're the same plant but different varieties. Okay, remember, much like animals, they have different breeds, but it's the same species. Remember our previous lesson? So, again, I'm showing you now, you culinary students or hospitality students or even the tourism students, oh, you got intrigued, diba? That these plants, these vegetables over there are actually the same species, the same plant. Okay? So, something to blow your mind. So, what is a genetically modified organism? So, according to dictionary.com, it's an organism or microorganism whose genetic material has been altered by the means of genetic engineering. Okay? So, how about a genetic engineer? What's genetic engineer? It's also called genetic modification, which is the direct manipulation of an organism's genome using biotechnology. So, what is biotechnology? So, I'm leaving you what's the definition of biotechnology. If you know the answer, what is biotechnology? If you found the answer to biotechnology, what is biotechnology? Comment in the chat box or in the comments below. Yeah. So para interaction naman tayo. Search nyo, Google nyo, ano yung biotechnology niyan. So how do you define biotechnology? Okay? So biotechnology, post your definition of biotechnology in the comments below or in the chat box if you're watching us live. Okay. Sige. I want you to give you a point of view on what are the types of GMOs that we are talking about. Okay. So there are 3 types of GMOs. We have transgenic, wherein a gene is moved from one non-closely related species to another. Second is a cisgenic or intragenic, which means it's a gen that is moved within the same species or closely related species. Then it involves what we call RNAi, which I will show you the video later on. And then the subgenic, na particular form of GMO, which is you edit the gene to amplify, delete, insert, or silence a certain particular gene in the genetic code of an organism. Okay, so I'm gonna show you the videos. I'll stop first the screen sharing, and if our YouTube algorithm is not so strict about what we're doing, so uh, allow me to let you watch the videos if we are allowed to do so. Anyway, I'll post the video on our MS Teams if it's okay with you. No, so allow me to insert the videos if I may be able to do so. Let's see if something comes out of our YouTube. So there are 2 videos that are in our let's just arrange them in. Sorry about that. Ganito talaga pag live. Hopefully the YouTube algorithm isn't angry about this. Let's see. Let's try to if and when YouTube detects this as something copyrighted or what, let it be. Okay. Let's watch this video over here. Oops.
Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different for mice, and then for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big. So big that to RNA researchers like Greg Hannon, RNA is more important than DNA.
DNA really works for RNA, and proteins really work for RNA.
Would you get an argument, by the way, from somebody else?
Oh, undoubtedly, sure.
So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and, well, we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code inside the nucleus. DNA, says Greg, never leaves the nucleus.
You ever meet one of those mean librarians, you know, the
Yes.
special reserve section?
The ones that go, pow.
Right. You can take the thing, you can copy it, but you can't take the book because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that's what RNA does. That scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm sea that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up in that sea, you see hundreds of thousands as well. We've made them into little guys with chef hats.
Those would be ribosomes, and in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, all cells. So all these chefs are basically building you. You are made of proteins. And because of RNA, we can copy, we can distribute, and we can cook up you and me. And RNA's been doing this for more than 3 billion years. But there was something spectacular about RNA that nobody knew till just a few years ago, and they learned about it, as we told you, by accident.
Here's a good one, maybe this.
In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower
That looks good.
to attract investors.
So that we could convince venture capitalists, investors, to give us more of the green stuff, more money.
Still, back in 1986, geneticists didn't know how to work that easily with, say, roses. And so
We began with a simple plant, regular garden variety petunias. Petunias being a plant that were easy to introduce genes to in 1986.
they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk, the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Completely white flowers. We lost pigmentation completely. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake?
A mistake. So we checked everything out, and there were no mistakes that we could find.
So why didn't the petunias turn purple? What happened?
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
And what was going on is, quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks. But some of those recipes, you'll notice, look a little different. And what's in these recipes is not good for the cell.
No, it's decidedly not good for the cell, because the sole purpose of that virus is to make additional copies of itself, and to the point that the entire cell is filled up with this, and the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape.
So when they see it in that shape,
They say virus.
And
They
they go,
say,
uh-oh.
uh-oh.
And the cop destroys the recipe.
and when you say it destroys, is this, should we think like a kung fu kind of thing? Is it like, hi, sort of deal?
Yeah, a little enzymatically, a little thermodynamics, things like that.
Enzymatically?
Enzymatic kung fu, maybe, yeah.
The cop destroys not only the oddly shaped version whenever he sees that recipe. Oddly shaped, regular shape, that recipe in any form must be destroyed to defeat the virus. And the interesting thing is, until 1998, nobody knew that cells had this defense mechanism.
We had no idea it was there. That's what's so amazing, is this whole mechanism had been sitting there where cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They'd worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The accident was Rich Jorgensen's purple petunia. The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cop saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell.
So there's no possibility anymore of producing the purple pigment because the purple transcripts are gone.
If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes in the cell.
And when scientists realized that every plant and animal cell has RNAi, a way to turn off the recipes, turn off genes, they thought, maybe we can use these cops to work for us.
Okay, Chubber?
Which brings us to Marty Russell, 78 years old. She and her husband used to spend lots of time here at their daughter's nursery.
Thank you, Rosie.
Years ago, she enjoyed doing lots of things.
Her passion was reading. She would read everything.
Golf.
Yeah.
I love to play golf, bridge.
But then Marty began losing her sight.
Couldn't see, and I'd probably get the peppers.
Before I continue with that video, if you have ever noticed something from that video, how viruses work, diba? Remember the mechanisms that are working behind the current pandemic? If you are ever observing the new scientific articles, all these readings about COVID-19, no? So basically it's basically what we say, a pirate invading a cell, attaching to a cell, then hijacks the cell by sending out RNA, codes that hijack the cell and then produce these poisons, and later on it overwhelms the cell, it destroys the cell. it should be like that, no? And with that note, guys, it can be said that um, the RNAi, as discussed, are like the cops. They are interceptors. They are interfering with these. These are the cells that. These are parts of our cell that makes defenses. They are these cells that are trying to check if the RNA being distributed in the nucleus or the nuclei are are right or are appropriate for that cell or for producing the right proteins, the right genetic materials. No, kind of amazing. No, and I don't know if I could continue with the video. If you want to continue with the video, I'll try to post the video in the description below. So baka copyright. So let's move on na lang to
with the
the.
zucchinis.
Let me na lang move on to the next video if it's not that long. No, if it's not that long. Let's see.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, boom. We are observing a video that um, goes into the study of how viruses invade our cells, and then how how scientists are trying their efforts to eradicate those viruses that can lead to disease. No, so we can see all of these great developments. No, and as you may notice, it's kind of what vaccines would work. No, just to give you an analogy in the current situation, some of these vaccines um, contain these particular genetic materials that are that are important to disable, or that are trying to disable, or either enhance the body's ability to detect the viruses that are invading the system, or trying to prevent the genetic code of the viruses to ever invade a particular cell, and somehow somehow disabling the work of the viruses. And through also the work of genetic engineering, scientists are now um, trying to establish or implement these changes to, or engineering the genetic code of the stem cells, or into the fertilized eggs, to somehow be able to succeed in creating organisms that are resistant to, say, bacteria, fungi, viruses, and other pathogens that can affect the way the organism works, or affecting the health of that particular organism. So that's really good. No, that's something for you to observe, especially now in our discussion, and the biotechnology of all, the study of genetic materials, how do they work, all of that. No, I hope you fully understood that particular notion. No, so again, as you saw in the previous two videos, the other one was actually long. No, but yeah, all of these are works of biotechnology, genetic engineering, genetically modified organisms. And somehow we should understand, why do we have to understand that? Because viruses exists. No, I just wanting you to let you understand why do these work. No, okay. So hope you are understanding that. Okay, so let's continue. Okay, so I'm showing a diagram of the difference between GMOs and genetically engineered organisms. This is a very large table. You might not be able to read this if you're not watching this in full resolution. But just to let you understand, how how genetically modified organisms work and genetically engineered organisms work. Basically, how we do like transgenic, cisgenic, and intergenic organisms, how these genes are being modified to be able to create a certain type of organism that has certain traits. So that's something we could answer. Anyway, you can watch this or read this through the slides that I'll be posting in your Microsoft Teams account. Okay. Some things to understand about genetic engineered organisms. on what are the traits that are being applied. No, first is herbicide tolerance that can withstand applications of certain herbicides, like um, for example, the Roundup Ready tomatoes or Roundup Ready organisms. So, what is Roundup? Roundup is a herbicide by Monsanto that is effective against certain herbs or weeds that affect certain crops like tomatoes, rice, wheat, other grains like that. No. So next is insect tolerance. No, that particular organism produces a certain toxin that could kill certain pests, like ano, like certain insects, like locusts, certain types of moths, other organisms or bugs that can kill a certain uh, particular plant. Also, it can also improve the nutrition of the organism to produce a nutritive value that is being changed or not to not produce change to not produce a certain anti-nutrient. No. Next is also resistance to diseases or stress tolerance. No, uh, being able to withstand low nutrient levels or excess nutrients that can be a result of weather patterns or certain situations that can affect the integrity of the crop. Increased storage life, it's also a trait of genetically engineered organisms. Also, medicinal value and also industrial values are also considered when producing genetically engineered organisms. Sorry about that. Right. So where are these GMOs being grown? So as a illustration, you can see most of them, for example, in the US, like innate potatoes. These are one of the most important crops in the world. Um, Innate potatoes are those genetically modified potatoes that when you peel them, they don't discolor. They are increased. They have increased shelf life. But they see decreased potential for producing toxins like acrylamides that are potential carcinogens when you cook potatoes at a certain high temperature, like when you cook them for French fries or for stews, and all of that. Now, when our uses of the potato. Um, and that's one of the breakthroughs of genetic engineering for these types of crops. Another example in Indonesia, the drought tolerant sugar canes. This is actually a public partnership with Ajinomoto for their production for MSG or monosodium glutamate, because MSG is really very aside from sugar, for of course, no sugar is also produced for vinegar or also for again MSG. So because of the high demand for MSG, Ajinomoto partnered with Indonesian Biotechnology to create drought tolerant sugarcane. No, this decreases the dependency of having to import sugar from outside. No, and and with that increases uh, the uh, the yield of sugar canes even in times of drought or dry season in Indonesia. I think I have known some uses also of drought tolerant sugarcane in Negros and also in Cebu, where they also grow sugarcane. And these have produced varied results, of success or failure. Sometimes um, some drought tolerant sugarcane, as said by certain studies, that they are also have the catch for drought tolerant sugarcane is they are not really resistant to certain weather phenomenon like typhoons that are prominent in the Philippines. Okay, so let's also mention the what they call the BT Eggplant, which is one of the pilot projects for Bangladesh biotechnology experts. No, this was the one of the uh, significantly important crops in Bangladesh, because in terms of their cuisine, and also um, more importantly, their applications also in various countries where eggplants, or what we call here in the Philippines, talong, how will they be significant in the country? So this is again one of the most nutritious and important vegetables. Uh, they integrated the genes from the BT, or Bacillus thuringiensis. No, these eggplants have been modified with genetic code from the Bacillus thuringiensis bacterium to decrease the ano to increase the resistance of these BT eggplant to insecticides, no, and surely to help the plants becoming resistant to diseases, no, placed by insects or pests. So this has been very successful in Bangladesh, but just to mention here in Mindanao, for instance, my home university, my alma mater, in UP Mindanao, had studies in relation to the BT eggplant, or BT brinjal, which were imported BT eggplants into the Philippines, which was opposed by no a lot of lay people. uh, s- Baka daw kasi karoon ng mga contamination, and so on and so forth, with non-native species, native species of talong, because of that particular research. This has been controversial actually here in the Philippines, specifically here in Davao and in UP Mindanao, when they were attempting to do research for BT eggplant. That was like the best, most discussed topic in our classes in biotechnology that time when I was a student in UP. So napaka-controversial niyan. Uh, um, Shout out to Dr. Eufemio Rasco, if you're watching, one of my professors before in UP Mindanao, who taught about uh, discussing biotechnology, and such a very what one of the best subjects that I have went through. Being able to understand why biotechnology has really grasped the the, the society in terms of crops that are important to certain cultures, certain nationalities. Such a such a sad story, no, na hindi masyado binigyang pansin, no. You can check this particular map in the link below. Can I'll try to put this in the description. Okay, what certain crops have a GMO trait now? No, what do these traits do? No, so for example, now there are certain types of crops that are resistant to insecticides, herbicides, and disease tolerant particular plants. No, for instance, let me zoom in. So, for example, cotton for insect resistance and herbicide tolerance, sugar beets, soybeans for herbicides, corn or maize for insect resistance, tolerance for herbicides, and for disease tolerance. No, um, um, herbicide tolerance for canola oil, for papaya for virus resistance, again certain viruses that could cause discoloration of papayas when ripe. Um, Alfalfa for herbicide tolerance, and also summer squash, or what we call ampalaya here in the Philippines, no, about for herbicide tolerance. So what can we say about that? It's all in these crops because of the work of scientists, because of the work of genetic engineering and biotechnology. Are you even afraid of that? Okay? So are you even afraid that these would potentially someday become a harm to us? I leave you with that. Examples also for genetically engineered crops that have been approved but not commercialized in the United States. For example, the conventional potatoes versus the innate potato, which I mentioned earlier. No, they are resistant uh, to blackening when you peel the potatoes. No, also Arctic apples. Do not discolor when you slice them and expose them to the air. So increased shelf life. But why are people not approving or not being commercial? Not why not these crops are being commercialized? Because there are certain ethical considerations to be done on here, no, which we can discuss in the next discussion in our next asynchronous session, wherein we'll be watching a certain documentary in which we are. I'll be posting on MS Teams for that asynchronous activity. We will also do a written discussion on that, written reaction paper of sorts. No, so in our last slide, even cheese for our for our tourism and hospitality students, uh, we have done our charcuterie plates or charcuterie boards rather, and we showcased our different kinds of cheeses and uh, fruits and um, other foods that are in a charcuterie plate paired with wine and all of that. All of these cheeses, some American cheeses are actually produced with a certain ingredient, a secret ingredient. Number one is rennet. It's a complex compound of enzymes that are found in most cheeses. No, uh, one of the key components of rennet is chymosin. It's an enzyme that curdles the casein or the proteins of milk. No, so with rennet, no, when you applied rennet to your cheeses, it prevents the curdling of proteins, and you have these very firm pieces of cheese, or sometimes soft. It depends on how you apply the rennet or the enzymes into your cheeses. No, so what that is really amazing discovery of how cheeses are actually made. It's still biotechnology, genetic engineering. So come to think about that. No, you culinary tourism and hospitality students, are you even thinking that your food that you are doing when you are trying to cook these foods for our culinary and hospitality students, are you even aware your foods that you are serving are actually GMOs? So I'm posing you that challenge now. No. So I'll leave you with that for our next meeting. And now let's do a discussion. How do you compare now genetically modified organisms with genetically engineered organisms? Are they the same? No. And do you think these organisms are harmful to our health and the environment? Yes or no? Tell us in the comments below. If you please answer this question so that you can earn extra points for your quizzes para at least interactive tayo. So again, how do you compare GEOs, genetically modified organisms, to GEOs, genetically engineered organisms? So, and do these organisms, both GMOs and GEOs, are they harmful or are they beneficial? What do you think? No? So let's lead it on that. So next meeting, we'll continue with our discussion on GMOs and gene therapy, wherein we'll also watch a documentary. No, I'll ask you to watch a documentary, and then you'll do a discussion reaction paper on that. Okay. So thank you so much, everyone. No, you're amazing guys, and hopefully by today, no, you are already preparing for your final project. So again, I'll be posting assignments, posting announcements for these, for these uh, discussion on GMOs, GEOs, and all of that, all of those sorts. So thank you so much, everyone. This has been Kenneth Forio. Thank you so much, and we'll see you on our next asynchronous session. Goodbye, and God bless you.